morning, dear students. Today we'll discuss physiology of synapse part 2. In the part 1 we have learned that basically synapse is nothing but a junction between two neurons. There can be different types of junctions anatomically like exonic, exodendritic and exosomatic, and physiologically there are two types of synapses. One is a chemical synapse and other is a electrical synapse. You will learn in detail regarding chemical synapse as it is dominant one. At only very few places we see electrical synapse where there is a gap junctions between the two neurons. So we will proceed with the properties of synapse. Just have an overlook. This is the nerve cell body showing cell body, dendrites, initial segment of axon and nucleus, nodes of Ranvier, Schwann cell, axon hillock, and these are the terminal buttons where there is a synaptic connections where there is a release of neurotransmitters. At a close look, cell body, so there these are different anatomical types at different places. Dendrites, you can see axon, junction of axon hillock and initial segment, and these are the axon terminals. So these are the different types of anatomical neurons. Now properties we just enumerate. The objectives of today's talk is different properties. First property is synaptic delay, 1 week induction, divergence, convergence, special summation, temporal summation, subliminal fringe, synaptic inhibition, fatigue, effect of acidosis or alkalosis, and effect of hypoxia. So these are the different types of properties which we'll discuss in today's talk. Let's see, there are different multiple synapses as we have seen in the first lecture. Neuron is single but they have got n number of synaptic connections. Like this you can see from the receptor, sensory nerve arises, ascending tract, and their connection with cerebellum at brainstem here. So at so many places, ultimately simply as I have mentioned you, CNS is nothing but neuron and synapse. Two things are there only. So there are multiple synapses are there at different places. You can see these are the sites where you have the synapse. Similarly, when the impulse is coming down in the descending tract, similarly we get multiple synapses at different places. So ultimately it is a play of only synapse at different places. At close look at the synaptic junction which we have discussed in the previous lecture. When the impulse arises at the synaptic terminal, there is voltage calcium channel gets open due to change in the potential, influx of calcium takes place which will take these vesicles near the site of release, and there is exocytosis of the neurotransmitter and which will combine with the postsynaptic receptors leading to generation of postsynaptic potential. So these are the basic steps. So ultimately the different types of neurotransmitters are the key which will have the effect on the postsynaptic membrane, and also the difference in the receptor. As I mentioned earlier, one transmitter is there but it will act on different types of receptor, will have different actions at different place. For example, very common acetylcholine, when it is acting in the heart, it is acting on muscarinic receptor and this leads to inhibition, heart rate reduces. Same acetylcholine acting at the intestine is have a different action, it will lead to increase in motility of smooth muscles. So neurotransmitter many times is same, it's based on the receptor, the actions varies. So this we have discussed last time. A presynaptic terminal, if there is a influx of sodium, this will lead to depolarization which will label as excitatory postsynaptic potential, and sometimes there is a opening of potassium channel, so there is efflux of potassium or influx of chloride. This will lead to hyperpolarization which is called as inhibition. So response is only of two types, either inhibition or excitation. So postsynaptic potentials are two types, excitatory postsynaptic potentials or inhibitory postsynaptic potential. So basically if you understand, CNS is in between these 3-4 things only: neuron, synapse, and in the synapse either excitation or inhibition. So by this way only we are going to understand different functions of nervous system. All whatever medicines we will be preparing for the nervous system will be ultimately having destination on the synapse and affecting the potentials. Either it is blocking the potential, or it is promoting, or it is antagonizing. Now the first property of synapse, as we know that in nerve impulses arriving at presynaptic terminal, and this ultimately leads to release of neurotransmitter from presynaptic terminal. It will act on here. So the conduction is, so there is a duration between all these steps. As we can see from first step, this is the seventh step. To take place, there's some time required. That time is called as synaptic delay. So this is the one property of synapse. So now if any reflex arc is having on a single synapse, that is called as a monosynaptic, it will have a short duration. But if reflex arc is taking too long, that indicates that it is having multi-synaptic connections. So through that also we can assess whether the reflex which we are trying to elicit is monosynaptic or polysynaptic. So if it is polysynaptic, we will have a more delay in the execution of that reflex. So this delay is mainly because of all the sequence of events which need to take place in this duration of time. So this is the first property called as a synaptic delay. So here you can see as the multiple impulses are going from here to there, and they're ultimately coming down from top to one, so there will be delay. Now second thing, as we know that sequence of events are from presynaptic terminal to postsynaptic, means unless until the neurotransmitter is released, they cannot be action on the postsynaptic membrane receptor. So that's why it can go in this direction only. It cannot go from here to there. If you excite this neuron, it will not lead to excitation here. Reason is that we need all the transmitters. So here there is no generation of neurotransmitter here. So that's why it is called as a one-way conduction. So if you stimulate a sensory nerve, impulse will go from the synapse to the brain. And if you excite a motor tract, it will come down to the muscle only. It will not go in reverse way. If it is going reverse, it will die here only. For example, if somebody, I'll show here. So this is one, this other. Okay, so if you are exciting this, what will happen? This will excite, go in this direction. So this neuron will excite here. But on the contrary, if you excite this neuron, impulse will travel here, it will travel here also, but it will not go from here to there. The reason is that we don't have this synaptic vesicles here. No, don't have the neurotransmitters. So what will happen? If you excite this, impulse will go from here to there only. So there is a conduction in this direction, not in this direction. Okay, so it will depends on this presynaptic terminal. So it is always a one-way conduction, whatever direction. If it is sensory, if it is a motor, whatever, it will go in one direction only. It will not go bidirection. Across the axon, that is bidirection, not across the synapse. If you excite an axon, so this is the cell body. So if you excite here, impulse will go in both direction, here as well as here. It's bidirection, but through synapse it will go to here only. It will not go from this to this. Okay, so this is the one. This is called this. So across synapse we are having a one-way conduction only. So here you can see, as I've seen, there are multiple neurons are there. You can see this is one neuron. This is one neuron, other neuron, third neuron, fourth neuron. So the direction of flow will be like this. So if you excite any of the neurons like this, so impulse will go across axon both direction, but it will not excite this, it will go here only. So that is called as a one-way conduction of a synapse. Now similarly, one neuron is there which will have multiple connections, especially in the descending tracts when we are coming from up cortex to down. So we'll have a multiple connections will be there. So from if you excite this, it will go to all like that. So this is called as a property of divergence. So the impulse will spread from one neuron to multiple neurons. This is called as the divergence. Similarly, there can be convergence also. Alpha motor neuron is there. On this, so many descending tracts will converge. We'll have a convergence of corticospinal tract. We will have a convergence of vestibulospinal tract. We'll have a convergence of oliospinal tract. So like this, so many inputs can excite one also. So this is called as a convergence. So there can be diverging circuit as well as a converging circuits can be there. So these are again two properties of synapse. If you stimulate one neuron, it will excite multiple neurons, and multiple neurons can lead to excitation of single neuron also. So these are the two properties, convergence and divergence. So here we can see one neuron, it is stimulated. You can see small potential develop, but not enough to have a fire. Not enough to have the fire. One more neuron is stimulated, presynaptic neuron. It will have a little bit more. So gradually there will be summation. So this is called a special summation. So if one neuron is excited, will not have a response. If two neurons are excited at a time, second neuron separately, we don't have a response, but two are combinedly, we will have the potential. So this is called a special summation of impulses. So this is again a property of neuron. If you excite multiple neurons at a time, we will have a response in the postsynaptic neuron. So that is called as a special summation. So here you can see practically. So here spin is being pricked. So only few neurons get excited. So this is one of the property of neuron also, that brain will property of decoding also, instant discrimination. So if stimulus is increased, more number of neurons excited. If it is more increased, more number of neurons excited, so more action potential generated. So this is again an example of special summation. Now similarly, if one neuron is excited, but excitation is continuous on that one, that will again lead to generation of action potential. That property is called as the temporal summation. As a train of impulses coming on one neuron will lead to firing of the postsynaptic neuron. So that is called as a temporal summation. Okay. So special summation is a summation of postsynaptic potential in response to stimuli that occur at different locations in the membrane of postsynaptic cell. Temporal summation is summation of postsynaptic potential in response to stimuli that occur at same location in the membrane of postsynaptic but at different times. So if you increase the duration, decrease the duration of stimulation, that will lead to temporal summation. These are the again two properties of synapse: temporal summation and special summation. So this can be explained by this diagram. Just simple diagram I have placed, X and Y. So this is the B neuron, A neuron. So if you A plus B excitation leads to excitation in the Y, that is a special summation, and X will be called in a stage of subliminal fringe. It's not firing but ready to go for the fire. So this property is called as the subliminal fringe. So if you excite A and B both, what will happen? As it is getting, Y is getting impulse from both, it will fire, but as X is getting only from the one, it is now ready to fire. So that property is called as subliminal fringe. Now very important property of synaptic inhibition, as we have mentioned, it is either excitation or inhibition only. Excitation is simple by release of excited neurotransmitter that will lead to generation of excited potential, but inhibition is of multiple types. One is simple inhibition, that is called as a direct inhibition, where there is release of inhibitory neurotransmitter, which will lead to either efflux of potassium or influx of chloride, leading to hyperpolarization. So this is called as a direct inhibition. Simple, but it is very simple. Now sometimes there is excitation by a neuron to the other one. There is excited potential, but other neurons are leading to inhibiting this excited neuron, just like putting up a brake on acceleration. So it is called as a presynaptic inhibition. So this excitatory neuron is exciting this postsynaptic neuron routinely. So routinely there is a excitation taking place across this synapse, but due to presence of other inhibitory neuron which are inhibiting the release of excitatory neurotransmitter, so they are putting up the brake on release of excitatory neurotransmitter. So it is called as a presynaptic inhibition. So it is very common, especially in the spinal cord at different places. So one is a direct inhibition, where there is a release of inhibitory neurotransmitter at the synapse, and presynaptic inhibition means across the excitatory synapse there is a brake, a neuron which is inhibiting the release of excitatory neurotransmitter. So it is called as one type of indirect inhibition. Now here you can see again excitatory neurotransmitter is there, but it is giving a branch to one more neuron which is inhibitory in nature. So here now it is called as negative feedback inhibition. Through this we are getting excitation, but through same it is stimulating this neuron also, which is now inhibitory in nature. So this is called as negative feedback inhibition. Okay, this will put up the brake on the constant excitation. Now one more type of inhibition, again across the excitation. So it is stimulating this inhibitory transmitter, but it is inhibiting something else. So this is called as postsynaptic inhibition. So here this neuron is exciting this, and this neuron is exciting this, which will lead to inhibition at the same place. So this is again different types of postsynaptic inhibition. So this is one more type. Now here you can see it is inhibiting directly this postsynaptic membrane. Initially we have seen in one it was inhibiting the here, but now in this case it is inhibiting postsynaptic membrane. So it is called as feed forward inhibition. Feed forward inhibition. Through this it is exciting, but through this it is inhibiting also. So these are the different types of inhibition. So normally what happens, you can see one neuron, so many inputs, so many conversions on this one from so many places. Some of them are exciting it, some are inhibiting it. So basically it is as per the requirement and order coming from the different sides. Sometimes from the vestibular apparatus impulse is coming, sometimes impulse coming from the cerebellum, sometimes impulse coming from the basal ganglion. So it is basically coordination of different parts of the brain which are controlling this excitation inhibition ultimately on the lowest neuron. Commonly it is the alpha motor neuron or gamma motor neuron. So like this there are multiple reverterating circuits are there. You can see here also. So parallel after discharge. So synapses are repeatedly stimulated, firing rate becomes progressively less in succeeding milliseconds or seconds. So exhaustion or partial exhaustion of store of transmitter substance in the presynaptic terminal, progressive inactivation of many postsynaptic membrane receptor, abnormal concentration of ions inside the postsynaptic neural cells. Now effect of acidosyl alkalosis, commonly in the blood, as if you see extracellular fluid is constantly maintained with the acid-base balance, but if a slight change in this will lead to change in synapse, which will lead to change in the activity of neuron, and which is very dangerous alkalosis greatly increases neural excitability. So it will reflect as seizure. So if the patient, suppose you will see a patient of seizure, simply check his blood acid base level. If you are finding that alkalosis, immediately change, give some antidotes or buffers, patient will get relief immediately. So if the patient is coming with epilepsy, you might think, "Arey, kuch tumor hai kya?" But if you see, if you analyze and do the ESF examination, if you are finding any change in acidosis or alkalosis, you will immediately treat the patient. So this can lead to comatose condition also, especially in diabetes mellitus will have. So this appears very critical condition. You can see the patient may be in the coma, maybe coming with seizure, maybe coming with so many problems, but the solution can be very simple. If you just do the blood acid base balance of the patient, and you are able to find some changes in this one, immediately buffers are given and the patient will be cured, will get relief immediately. So this is very important. If the patient is coming with such problems, we have to analyze and see what problem exactly he is having. Similarly, hypoxia is a very critical condition. If there is a decrease in oxygen deficiency, the patient may have the seizures, he may come with the coma, with so many problems. But simply give you oxygen immediately, patient will come down immediately, come up. So within 3 to 7 seconds, person becomes unconscious if it is having hypoxia because of any reason. Maybe simply choking in the airway, maybe because of decreased oxygen level. If you are going in the high altitude on some timber train, suppose somebody is going with a timber train, and if you get unconscious, simply give oxygen, the cylinder, patient will come back immediately. So like this, today we have discussed property of synapse, synaptic delay, one-way conduction, divergence, convergence, special summation, temporal summation, subliminal fringe, synaptic inhibition of multiple types, fatigue, effect of acetylcholine alkalosis, and effect of hypoxia. So here we are completing properties of synapse. Thank you. In the next lecture, we will discuss physiology of tracts.